Hey, what's up, Canada and beyond? How are you amazing folks this afternoon? I want to welcome you all and thank you for tuning in to what is going to be the first part of, I think, about 14 different doctors that we are lining up for a series called Doctor Talks. And with all of the inundation, with everything that everybody is seeing and all of the noise out there about everything going on from every level of measures and this trying time that we're in, we're doing our best to bring some of the experts that can help us cut through the noise and speak to their expertise to you guys and hopefully provide some clarity for everybody moving forward, that the people are going to be able to make educated decisions. So this is the point and focus of all of this. So today, our first guest, I would like to welcome you guys and introduce everybody to Dr. Steven Pelech. He is an expert in his field. I couldn't begin to rattle off his credentials or even probably understand them, as most of us here are like this. But I mean, this doesn't mean that we're oblivious to what's going on. So welcome to the show. Thank you for taking the time to join us today and give some of us lay people out here an idea of what your expertise is and why you are so qualified to do this.
Thank you, Wayne, and hello, everyone. I appreciate the opportunity to tell you a little bit about what it is that is going on in my university lab, University of British Columbia, and with my company, Kinexus Bioinformatics Corporation. My background is that I have a Ph.D. in biochemistry, which is obtained through the University of British Columbia. I trained for 5 years after my doctorate degree in the United Kingdom and in the United States, working in the lab of a Nobel Prize winner, Dr. Edwin Krebs. And then I became a scientist at the newly formed Biomedical Research Center at the University of British Columbia. And I've been on faculty for about 32 years now as a full professor in the Division of Neurology in the Department of Medicine. And I've also founded 2 biotech companies, one of them called Kinexus Pharmaceuticals. I was the president and CEO of that for 6 years and its chief scientific officer. And then I founded another company, which I'm also running now for the last 21 years. It's Kinexus Bioinformatics Corporation, located in Vancouver. And most of the research that I do is actually focused on looking at the cell signaling systems that operate inside living cells that allow cells to respond to their environment. And defects in these systems can give rise to hundreds of different diseases, including cancer and diabetes and Alzheimer's. But in the last year, because of the whole COVID-19 pandemic situation, I felt that some of the technology that we had in our company was actually ideal to use to more explore what is it that people are making antibodies against in the case of the SARS-CoV-2 virus. Now, I should qualify everything that I say, that what I'm going to tell you is my opinion. It does not represent the University of British Columbia. There are others at UBC that have similar outlooks and others that disagree. So bear that in mind. There's a conflict of interest disclosure that I am the major shareholder of Kinexus Bioinformatics Corporation, and the company does sell products. We don't sell any products related to COVID-19 at this time, but as you'll see, I'm looking to develop a test which will at least assist in understanding what is going on with the SARS-CoV-2 virus, which is the virus that causes COVID-19. And I'll tell you a little bit about what it is that we're doing. So if that's okay with you, Wayne, I can describe.
Absolutely. And before you get into the kind of the meat and potatoes of this, I'd like to highlight the significance of this is that, this is meaningful discourse. This is what you're doing and being upfront and open. And we appreciate that not everybody agrees with everybody out there. And this is okay. This is what we need to have meaningful discourse moving forward in all of this, because this is such a new situation that the world is finding itself in. And we're not seeing the things in the real world that justify everything. And you guys are looking to the root of the matter in this and to find answers that make sense for everybody, in spite of possibly at times less than comfortable discussion. This is significant that you're taking this responsibility to speak to the public. Thank you.
Thanks, Wayne. So I've published about 240 scientific papers in peer-reviewed journals over the last 40 years. But my research in the last year has been quite focused on COVID-19, in particular the SARS-CoV-2 virus. And one of the reasons why we decided to go down this route was because of unique technologies that we have in our company that allows us to essentially take the gene sequence, the genome of this virus, which was worked out basically last year around January. And when it became available, a gene is like a blueprint to make proteins typically. And RNA, which you may have heard of, is sort of an intermediate copy that can be made several copies of that original blueprint, which is the DNA gene. And this is, of course, the RNA is the genetic material that's being put into, let's say, the Moderna or the Pfizer vaccines that people are taking. And from this RNA, you make the protein. The RNA is like a blueprint copy that's used to instruct the protein synthesis machinery inside living cells to produce the virus, in this case. So normally these ribosomes, which are the protein factories that make more proteins, they actually are producing the normal cellular proteins. But the virus, when it infects cells, it basically takes over so that now that machinery is used, molecular machinery is used to actually make virus proteins. And that's how the virus replicates. Now, the genome of this virus is about 30,000 nucleotides long. Nucleotides are like letters, A, T, C's, and G's, that specify the order of the amino acids, which are the building blocks of the proteins that are found in your normal cellular proteins and also in the virus proteins. So there is a small number of proteins in the genome that are actually specified for this SARS-CoV-2 virus. And SARS-CoV-2 is a coronavirus. These viruses are actually very common in the environment. Many of them cause common colds. And remarkably, many of those same proteins that are present in the SARS-CoV-2 virus are actually present in these other common coronaviruses. They're not exactly identical, but they are very, very similar in their overall structure, and they work very much the same way. So, for example, the SARS-CoV-2 virus can kind of look like a ball that has sort of a lipid membrane, a fat membrane around it to contain it like a bag. And inside it'll have some virus proteins. But on the surface of the virus, it'll have these proteins that extend out. And the most characteristic of these is so-called spike protein. It's the largest protein. And the spike protein has the ability to latch on to normal cells by interacting with a protein on the surface of those normal cells known as ACE2 for angiotensin converting enzyme 2. Anyways, The important thing is that the spike protein attaches to this like a key binding to a lock, and it allows the virus to enter inside of cells. And there's a few other proteins on surface of cells that the virus can also attach to, CD42 and neuropilin in the brain, and CD4, which is on T cells, for example, which are lymphocytes, white blood cells, your immune cells. Anyways, we could take, with our technology, we can artificially make these proteins of any structure that we want on membranes, so that we can actually recreate the entire virus proteome, the structures of all these proteins from that virus, on a little membrane. And we can do hundreds of different pieces. What we have actually done collectively is looked at the entire structure of the virus as predicted from its gene sequences, the protein structure in little pieces. Now, these little pieces, we've dissected the proteins of the virus into 8,000 separate little pieces that have some little bit of overlap between each piece, but basically we've tiled out the entire structure of the virus, built it artificially, and with this tool that we've created, we can now take blood from people who have recovered from SARS-CoV-2 infections, survivors of COVID-19. They have antibodies that they have produced in their blood to be able to fight the virus and eliminate it. So these antibodies, they're also called immunoglobulins, IgG, IgM, IgA. These are three classes of these antibodies. They exist in your blood, they bind to the virus, and they take it out. And so we know that people that have recovered from the disease have these antibodies. So the question is, which part of the virus are people making antibodies against? And if we know which parts that are the vulnerable parts now of the virus that our immune system is attacking, then we could in principle take those pieces and inject those pieces into people, and they can make antibodies against the virus. Now, this is something that our company has done for the last 21 years. We actually take and artificially make pieces of the virus or proteins inside the body that we want to study for cancer research or diabetes, and we inject those pieces into rabbits under their skin. And after a matter of weeks, they make antibodies against those pieces. And we've made about 1,600 different antibodies this way very successfully. And we know that if we put these pieces into people, they would make antibodies against these pieces. And those antibodies would recognize, they would bind to the actual proteins that are the complete intact proteins. And that's how a lot of biomedical research is actually done today, to study how disease processes occur and to identify biomarkers of disease. So this is not unique technology in terms of making antibodies in laboratories, in animals, and in people. So nothing new about this. Now, with this approach, there's two things that we've really learned. Firstly, when people are infected with exactly the same virus, their antibody response in terms of which parts of the virus that they make antibodies against is completely different amongst people. So out of those 8,000 different potential parts of the virus that you could check to see if people are making antibodies against, we narrowed it down to a few hundred. In fact, even down to about 100 that are the most commonly observed as being what we call immunogenic, that is inducing an antibody response amongst all the possibilities in the virus. So these 100 best target sequences of amino acids and these peptides that these antibodies are being directed against, again, they differ from people But for people who've recovered, they'll have at least some of these antibodies that they will be very successful in producing that will recognize and lock onto the virus. So the second major point to understand, apart from the very differences in response of people to the same virus, is secondly that those different antibodies that you have, they only last for in your circulation at best about 21 days. So if you are not challenged with the virus, then those antibody levels will decline over time in your body. Now, that's okay, because the cells that produce antibodies, we call these B cells or lymphocytes, B lymphocytes. These immune cells, they go dormant in your blood system. And sometimes they're called plasma cells, but these dormant antibody producing cells can live for years in your circulation. And when you get exposed to that same virus protein that induced the antibody response in the first place, those B cells will become reactivated and they will start producing antibodies, and those cells will divide and produce even more of these B cells, produce more of those antibodies. And so you get a very strong immune response in time that by the time the virus starts to get inside you and replicate, the immune system has already been alerted and activated and is producing antibodies that are neutralizing that virus and taking it out. So you have that protection. And we call that immune memory, but it does take repeated exposures to really establish that immune memory. And this is where one of my concerns relates to the vaccine that I'll discuss in a few moments. So the other important thing to remember about when you have an immune response, and it's against a virus, for example, the virus has many different proteins. The spike protein is only one of them. Like I said, it's the largest protein and really sticks out. Hence this coronavirus is the name. Corona means crown. So the virus looks like it's got a crown around it.
There's a lot of animation, visuals of what this looks like. I think most people are pretty. It seems to be at least uniform in how they visualize what this looks like.
That's right. And so what happens is that the spike protein is really what's being targeted by vaccine companies, and it's a logical place to try to attack with antibodies. But there's actually other proteins on the surface of the virus as well, a membrane protein, a envelope protein, and then there's proteins that are inside the virus. Now, practically speaking, antibodies that target the proteins inside the virus are not going to be as helpful as the ones that target the outside of the virus, because that's what's exposed to your immune system. And the key thing to remember is that when you make antibodies, you're not making it against just one part of the virus. You're actually making it against many different parts of the virus. So one of the things that we've learned from our research is that, yes, you make antibodies against, every person makes different antibodies against different parts of the virus. There's certain areas that tend to be more common amongst these people, but you have a whole repertoire of different antibodies targeting different parts of the virus. So when you have a mutation, and there's a lot of talk about these variants of concern, where you have these mutations in the structures of the SARS-CoV-2 virus. So you have these variants that are coming from the United Kingdom or from South Africa or Brazil. When you actually look in the spike protein and some of the other proteins, these are very, very small changes in the overall structure of the protein. So when we have a mutation, remember I mentioned 30,000 nucleotides, you only need to have a change in one nucleotide, and that's considered a new mutant. But when you're talking about the overall structure of the proteins encoded by those genes, they're 99.5% the same as the normal virus that originally caused the problems in the first place. So your antibodies, they may, in some cases, a few of them might recognize that particular mutation, and with that change, those antibodies don't work. But 99% of the antibodies you have still should be working just fine on mutant strains of the virus. Now, it's very peculiar that there's many reports that some of these vaccines that we have now that are being utilized don't seem to be as effective in a number of people with these mutant strains. And it's, as an immunologist, which now I have an immunology background, I find that very difficult to explain, as do a lot of my colleagues. So I think there's more that's going on there that meets the eye, that, you know, maybe the people, these resistance, I'm not sure that maybe the vaccines were even working in the first place. So recall, I told you that when everybody is exposed to exactly the same virus, they have a totally different antibody response. Well, that will be true also for the vaccine. So the vaccines that we have actually target the spike protein only, okay, not the rest of all the other viral proteins. So already your antibody response is going to be much more narrow in where it can actually develop antibodies against. Coupled with that, knowing that people are very different in their immune response, you're going to get people who have been vaccinated who actually do not have an immune response that's going to be protective. Now, what do I mean by protective? Well, ultimately, it's whether or not you're going to become successfully infected and get sick with the virus. But the way the body works, the immune system is very complicated. It has a number of different cell types that are at work to protect you. The B cells that produce antibodies is just one arm of your immune system. You have so-called T cells. These cells seek out and recognize viruses and bacteria to destroy them. You have dendritic cells that are part of that process. You have phagocytic cells like macrophages. So for example, in your lungs, 20% of the cells in your lungs are macrophages that are cleaning up that environment. They're eating the bacteria and viruses and other particles, and then they break it down so that it's inactivated if there's any of these bacteria or viruses. So you have a large number of these cells that are protecting you of different types. And bear in mind that a white blood cell, an immune cell, is about one of those for every 400 red blood cells that you have carrying the oxygen through your body. So they're a small portion of the cells in the blood, but they're very active in what they do to protect you. And so these different cell types, they're working with each other. Now, if you have antibodies, what we're testing right now when we say that an antibody is neutralizing, most researchers are really talking about whether or not that antibody is binding to the part of the spike protein that allows it to bind to the ACE2 protein to get inside cells. So the idea is that if an antibody that can lock on to the virus protein, the spike protein in that region, then it kind of barricades it so that it can't get inside cells. And so we call those neutralization assays. And those is one measure of how you can be protected by antibodies against the virus. But in reality, any surface of the virus that in the intact virus that an antibody can stick to actually tags that virus so that these lymphocytes and other phagocytic cells that engulf and eat and digest the virus are able to actually process it and destroy it. And doing so, what they actually do is when they digest the proteins that are in the virus into little pieces, they then present those pieces in the context of these proteins that are normal cellular proteins that when they're presented with these pieces, then signal to the B cells to make antibodies if those B cells recognize those pieces. So it's part of an integrated immune system that provides you with protection. And even though you have no antibodies, let's say, you still have these T cells that are activated that will seek out and destroy the virus too, because the T cells have the capability of recognizing specific sequences that will be from these parts of the virus protein. So you have a fairly broad immune response. So in general, someone who actually becomes infected with the virus has a multi-pronged approach to actually take on that virus should it arrive again in the future. And you'll get a better immune response to the actual virus than you will to the vaccine, which is just for a portion of the protein.
And that's being the kitchen table guy. That's kind of what I'm hearing out of this. And I really hate to simplify it, but for the average person, it's like that's neck deep in building and working and living their daily life. It's a bad bug, but it's really not worth being bombarded by the discussion of all of these variants and all of these things when our natural immune system is probably more effective on this bug, that we have a 99 point something survival rate, and this vaccination that they're issuing out there, although it's 4 different brand names on it, really are kind of targeting one thing that our system is probably doing already anyways. Other than, obviously there's going to be some people that have different environmental influences and medical and health issues that are going to react differently. But in general, for the average person, this is a lot of hoopla about things that are pretty natural to them.
Yes, this is what's kind of annoying for someone like myself, where there's such a foundation of 50 years and more of modern immunology where we've learned so much. And it's like when this virus has showed up, we've kind of discounted that and say, well, this is a whole new virus and we don't know what it's really like. But the reality is we've dealt with coronaviruses in the past and we know basically how they work. What we are probably in greater lack of knowledge now is in terms of the vaccines that we're using to treat the virus. Now, don't get me wrong. I think this virus can be very deadly, especially in elderly people and people that have complications of obesity, smoking, diabetes, cardiovascular disease, pulmonary disease, immune deficiencies. In this case, it's actually, if you're at high risk, I recommend the vaccine, especially for elderly people. Where I have a caveat is people under age 50 actually getting vaccinated. And the reasons for this are multifold. One, all of the vaccines are experimental vaccines. They have not actually been received what we call completed phase 3 human clinical trials. Any drug or vaccine that is normally produced has to pass phase 3 human trials where they've been well tested for efficacy, that is how well it works, and safety. And the testing that has actually been done on these viruses is being done on the general public with hundreds of millions of people being vaccinated with products that have actually not yet been formally reviewed as we would normally do for any drug that is normally given to people or vaccine. So, sorry, Wayne?
And this is exactly what the average person is noticing and is not really okay with. And I got a chance to speak with you for a moment before the show, is that most of the people that are looking for answers, they're not necessarily anti-vaccination, but they are accustomed to a long, thorough process in the treatments that become available to Canadians. And as you alluded to, you guys are working from 50 years of research, and yet we are seeing our current governments completely abandoning this long established process that is based on actual science and facts and information, and you guys have got all of this, and you guys Canadians are seeing that you guys are kind of being shut out from the discussion from these things that are trial and completely have no long-term data for. So this is I think a natural reaction people should be having to something when they're seeing this kind of pressure societally, mounting on this subject. So I just kind of wanted to touch on that a little bit, that, this changing of process is incredibly alarming.
That's right. And apart from the fact that it's an experimental vaccine, that we do not know, firstly, how long does it actually last? We know that there is good evidence at this point that if you follow the proper protocols as recommended by the vaccine manufacturers, that you can get effective protection for at least 4 to 6 months. However, this is based on the idea that you have, in the case of the Moderna and the Pfizer vaccines, these RNA vaccines, which, by the way, this is the first time that this technology has been used to make vaccines to fight against an infectious disease. They were originally tried to be used for cancer therapies. And again, it's still early stages for that approach. But what we've been doing in Canada, because of the shortage of these vaccines, we've been extending the period between the booster shot and the initial injection. So the manufacturers recommend about a month, and then you should have your booster shot. Scientific papers
with a treatment that is already questionable and without answers, they're already changing the game and the rules on the fly as they go to cover up their political ineptitude.
Right. The government is basically doing what is not recommended by Health Canada. They are doing what, from the scientific papers that have been published, shows that actually some of the neutralizing activity drops off after a month after the first injection. Antibody levels can stay high for a bit, but then they will decline. And so going with a 4-month period between the initial injection and the booster shot is certainly not what the manufacturers recommend. It's not what they tested for. It's not what any scientific papers that I've seen have supported. And frankly, I'm concerned that people who do get the vaccine may have a sense that they have protection, that perhaps 2 months later, they don't actually have protection. And then there's another 2 months before they get their booster shot. And if you don't have a re-exposure, then what will happen, like to the virus, then what will happen is you will not establish immune memory. So those B cells that I talked about that would be there to protect you in the future, they don't really get a chance to get established. And so what would happen is after the second injection, 4 months later, it'll be just like you had it the first time. So then you should have another booster shot a month after that. And so some people will get protection. Now, the good news is, and I haven't mentioned this yet, but when we actually have been testing people with our tests that we developed to see whether or not people are developing antibodies and which parts, we actually tested a lot of healthy people, people that never had any symptoms of SARS-CoV-2. Some people would, if you listen to different sources, the argument is that very little of our population has actually been exposed to the virus yet. And we've got to protect them from getting exposed to that virus. But when I do my tests, and not just with our technology, but with another technology produced by another company, mesoscale devices, we got some pretty astonishing results. We found, and this is published, by the way, peer-reviewed, published work I'm talking about now, done with Pascal Lavoie, Dr. Lavoie at the BC Children's Research Hospital, using the so-called mesoscale devices antibody test. They determined that out of 276 people that they tested, that were healthy people, no prior indication that they had COVID-19. They weren't sick. Half of them were healthcare workers. We found that 90%, 90% of the people that we tested had antibodies that would recognize the SARS-CoV-2 protein in the Vancouver area.
So now let's specify, be clear that this is not the PCR test. This is the technology that you guys have been developing.
Well, this is not even our technology. This is the technology used that's being used in the States and in Canada. It's very sensitive, and they only test whole proteins. They don't test pieces. So they test a variety of cold coronaviruses, the ones that cause common cold, and SARS-CoV-2. And basically what we found is that these people all had antibodies. They would differ in which antibodies and parts they would have. Now, the initial interpretation of the results was that, well, it must be that when people were exposed to these cold coronaviruses, that the antibodies they're making against those are recognizing the SARS-CoV-2 virus that causes COVID-19. And that, at first blush, seemed a reasonable hypothesis. But then we took 50 of their samples, of these people, of the 276, and we tested them with our test. And we not only confirmed the results for the spike proteins that were being tracked with that test, but we also confirmed it with antibodies against the other SARS-CoV-2 proteins and in the different pieces of the spike protein. So we confirmed and extended the results of that study, and that has been published. Meanwhile, in our own lab, we've tested about 240 people now, of which about 70 of them are bona fide SARS-CoV-2 patients. They tested positive with the PCR test, the genetic test. And we're finding that the vast majority of those people also have antibodies that are healthy. So the question is, well, how can this be? Because when we start looking at the pieces and looking at how they compare with the corresponding parts of the cold coronaviruses, they are not that similar in those parts. So that means that it's more likely the antibodies that we're seeing are actually against the SARS-CoV-2 virus itself. So then the question is, well, how can that be? How can there be such a widespread number of people that have antibodies against the virus? And of course, the important question is, are they protective? And we haven't published the data for that yet, but we certainly have evidence that the blood samples from these healthy people are able to neutralize the ability of the SARS-CoV-2 virus itself to infect healthy cells in cell culture in experiments and dishes. So they do appear to be protective. Now, how can this happen? Well, the narrative right now, and I'm based in British Columbia, is that the BC Centre for Disease Control, their basic message is that, well, the virus really started here in Vancouver area sometime in early March, and it principally came from Europe. Now, anybody who lives in Vancouver, and I live in Richmond, which is a suburb of Vancouver, 57% of the population of Richmond is actually Chinese. And about a third of the population in the Vancouver area is Chinese. And so a lot of these people are recent immigrants, and they're fairly wealthy. And so they travel back and forth with their businesses back in China. So there's a huge amount of back and forth travel. And we know that the virus was certainly spreading around in Wuhan back in October and November of 2019. And if you look at the restrictions that happened in the Vancouver area, they were really introduced not till about the middle of March. And actually, we did very little testing in BC. We had the lowest testing rate of the provinces for the genetic test. And if you were sick, they just told you, well, unless you're really ill, assume you have COVID-19 and stay home. So we don't really know what the size of the first peak was in the Vancouver area, certainly BC, and it's a dramatic underestimate. So for probably, we have tested people that were ill with COVID-like symptoms in December of 2019. We could measure their antibody levels 6 months later, clearly. And many dentists we checked in January and February that had COVID-like symptoms, and we confirmed that they had antibodies. So we know that the virus was spreading around in the Vancouver area for at least 3.5 months before there was any restrictions.
It's interesting you say that, because I was pretty sure that I had experienced a flu kind of worse than anything I had experienced in 20 years, and it kicked me around for about a week, but I got over it, and I've never had any issue since. And I'm ordinarily a pretty healthy guy, so it was. I mean, you expect these things from time to time, and hopefully I'll be able to get you to point me in the right direction for that paper that you described there earlier. There's going to be a lot of people that would like to see that, and any future papers that you publish, we're going to be paying a lot closer attention to the work that you've been doing. Why do you think we're not hearing discussions like this? Why are we not hearing from this stuff, what you're talking about, from the public health authority? Because what you're talking, even from the level of knowledge and expertise and training that you have, seems pretty straightforward to us in the audience, and it makes a lot of common sense. And we're not hearing any of this from any of our health authorities. It's actually quite the contrary.
Yeah, I think people are doing the best they can. There's a lot of researchers working in this area, and it's new to a lot of people. And I think there is a overcaution, the idea being that, well, people have died. And here in British Columbia, about 1,600 people have died from this virus. But unfortunately, things are not put into context. So for example, we know that 100 people die every day in British Columbia from all causes. Mm-hmm. and most of them are very elderly. We know that of those, in the last year, for 2,000, about 3 people have died every day in BC from COVID, if you average it out. Now, what's interesting is if you take the number of people who have died from illicit drug overdose in 2019, it's about 3.5 people per day. But if you take now 2020 and you look at that year, there's a 75% increase COVID associated with illicit drug overdose. So that's equal almost to the number of people that have died. Like about 6 people die every day now in BC from illicit drug overdose. It's almost the same number of people that has actually died from COVID-19 in British Columbia. And I'm not talking about the total number that have died from illicit drug overdose. I'm talking about the increase associated with COVID, where when you have these restrictions, you create anxiety and depression and substance abuse. And with that increased substance abuse, the greater opportunity to have these illicit drug overdoses. And so you have to put it really into perspective. And the average person who's died in BC of COVID-19 is actually a couple of years higher than the average life expectancy in the country.
I've seen the same thing in Alberta. I'm an Albertan at heart, born and raised there, and the average median age of death has actually increased a year this year. The actual number of deaths in Alberta from all causes is the lower in 2020 than it was in it was the second lowest year in the last 5 years. But at the same token, the abuse injury, the suicide rates, especially among youth and teens, everything else is off of the charts. And myself, I spent 15 years in fire and rescue as a frontline worker. So I see trauma and I recognize trauma. And this is what I'm seeing everywhere I go. People that are they're stumbling about their daily lives in a state of trauma, literally. even if it's not a physical trauma, it's become an emotional and mental trauma to people. So this is what I'm seeing as a byproduct of, or ripple effects of what is going on here, and why I'm so passionate and committed to giving folks like you voice, so we can try and nullify some of that and inform people.
Well, here's the good news. Like I said, it seems as though this virus has actually spread around quite a bit, and a lot of people already have antibodies and aren't even aware of it. So in fact, most people who are infected with this virus, especially if they're younger, like less than 60 years of age, most of them are asymptomatic. They didn't even know that they had it. Secondly, people who do get it, in most cases, it's fairly mild. It's a very, very small percentage of people that need to be hospitalized and that would actually die from it. So it's not to be taken lightly that there is this death and that you need to be careful. But on the other hand, we've seen certainly over the last, since Christmas in British Columbia, the death rate has just plummeted in BC, as it has actually in many of the other provinces. And the reason is that a lot of the elderly people well, I should point out that the drop has occurred before the vaccination. Okay, so we have this process where those that are vulnerable have probably passed on, and the people that are left are the survivors. So if you were over age 80 in BC a year ago and you got infected, you had a 1 in 4 chance of dying from COVID-19. But if you're 50 years or younger, the chances of dying from COVID-19 was 1,000-fold less. Now, the chances of dying in BC, if you're elderly, is actually about the same, for an 80-year-old and older, or a 70-year-old or older, as a 60-year-old or older. So the death rate has actually come down, and I think that's been helped in part by the vaccinations in that population, but some of it is just natural selection over time. People that are vulnerable will pass on, if not from the SARS-CoV-2, then probably some of the other infections. Because the average, like I say, age of someone who's dying in BC right now, COVID-19, is about 84 years of age. So that's one thing that's going on. But the bad news is we have more people that are ending up in emergencies in hospital, and they're younger people. And the question is, well, why is this happening? Why didn't we have young people a year ago being a major portion of the people that are in ICUs or just going to emergency and hospitalization? Why is it all of a sudden really spiking up now? And to me, when I look at this data, it's indicative of a much, much bigger problem that's at hand. So there's two things. One is I'm worried about the vaccines being experimental, that we don't know what the long-term effects are. I think if understanding the mechanism of action of how does these vaccines actually work. So if we take a look at the Moderna or the Pfizer vaccines, these RNA are delivered in basically a lipid bag that fuses with mainly muscle cells. And the muscle protein synthesis machinery takes that RNA and reads it and makes the spike protein, and then it sends it to the surface of the muscle cell. And on the surface of the muscle, it remains attached. It's tethered. It doesn't just float away from the cell. It's actually stuck on the surface of the muscle cell. Now, as I mentioned to you, we have antibodies against other common coronaviruses that can cross-react, or they can also recognize SARS-CoV-2, including the spike protein. So what happens is you actually get antibodies sticking to the surface of the muscle cell, and it initiates an immune response where the immune system now attacks the muscle cells. Now, I think in the mechanism of action of the vaccine, that's probably how it's working. The destruction that's occurring on the muscle cells allows the release and the eating of these proteins, including muscle cell proteins on the surface. presumably the muscle cells are surviving the immune attack. We would hope so, because in your body there's only two types of cells that don't divide and renew themselves. It's muscle cells and neurons. So the immune response that you're getting is those lymphocytes are coming and they're attacking and eating these proteins and then presenting them with their major histocompatibility antigens for the B cells to make more antibodies against. So I think this is the reason why when you first get the vaccine, the symptoms are pretty mild. But when you get the booster shot, it's much more severe and dramatic. In fact, it's the same symptoms as you would get from the virus itself in many cases. So that means that you could be setting yourself up in the long term for autoimmune disease, because with that antibody response, you could potentially break what we call the problem is when you have antibodies that recognize your own proteins, your own cells, you create an autoimmune disease. Now, this does not show up very fast. It actually takes a long time for people to acquire autoimmune diseases. But if you give the vaccines to very young people, then there's a very high chance that over the course of their life, they may develop that autoimmune disease as compared to someone who's much older. So that's one concern that I have. There's other evidence that that the spike protein itself may be interacting with cells that's like blood cells that could cause blood clotting, and platelets in particular. So there is evidence that we have increased thrombosis associated with a very, very small number of people who are actually immunized. In terms of tracking adverse vaccine events after these vaccines initially, it's very rare. Only about 5 out of 1,000 people will actually end up with an adverse vaccine reaction that's immediate. But we don't know what the long-term consequences are at this point. So when you put everything in balance, we know that people that are younger are at very, very low risk of actually dying from SARS-CoV-2. In British Columbia, until recently, we had 0 deaths that were under 30. I think right now, when I look at the latest figures of this, only about 8% of the population under age 50 that gets COVID that that there's been deaths in the province that have been hospitalized. So it's a very, very low percentage of people that actually, if they're immediately vaccinated, will have an adverse reaction. But then again, the problem with some of the reporting for these adverse drug reactions is that they're dramatically underreported in many cases, because it's self-reporting that's required. And a physician or someone has to do it. And if there's other complications, we don't know whether it's COVID related or vaccine related. In some cases, people are testing for COVID, but we're not testing easily for whether or not it's the vaccine that's actually caused the problem. That information will come as time goes on, but there's enough there that it makes no sense to vaccinate people that are extremely low risk, that probably aren't going to have symptoms or will be mild, and has very low risk of actual death. And so you can say, well, but don't we need to vaccinate these people so they don't spread the disease to other people that are more vulnerable? But at the same token, if we're vaccinating those people that actually are at high risk or elderly, then this way we can still establish herd immunity for people that are in the general population that are healthy, so that they'll be better protected against the coming mutant strains of this virus in general. So I brought up that question. Why is it that so many people are going now that are younger to emergency in the hospitals and being hospitalized? And I have a theory. It's a theory. And it's 3 factors, I think, that are at play here. Number one, younger generation tends to be less social than past generations. They like to hang out, usually in their bedrooms, socializing through the internet. So there isn't that in general exposure, except if they're going to school or some other environment where they could have that opportunity to get together and be exposed to general viruses and bacteria in the environment. Now, I should mention that there's at least 350,000 different types of viruses that are in our environment that affect mammals, and they can hop from one species to another. So you can quarantine all of the people you want, but these animal vectors are still there that coronaviruses can spread. Just with SARS-CoV-2, you can have it in pangolins, you can have it in bats, you can have it in minks, people, cats. So all the quarantining you want or vaccination you want is not going to help when you've got these animal vectors. Now, the second thing, apart from the fact that these younger people are not getting exposed to these viruses and educating their immune systems, with all the lockdowns, the more that you lock down and quarantine people, and the population has been really quite, I think, compliant, including young people, wearing masks and watching out for their elders by being careful with their activities. In fact, we've been so good at this that I remember in BC looking this up for the flu season this year. There's only been less than around 10 or so cases of influenza in BC, whereas normally about a third of the people that are tested have antibodies against influenza. So, and by the way, those cases of influenza are from influenza vaccines that were given to those people. So we know that the protective measures that we're taking are extremely effective. Now, what happens when you do that over a prolonged period of time is that your general immunity against not just SARS-CoV-2, let's say you had been infected and you've got antibodies, well, that's going to decline if you don't get exposed to the virus again. But also your protection against all these other bacteria and viruses that are in the environment. We know that if you had a vaccination against influenza, or you had a vaccination against BCG, like for tuberculosis, that you're actually protected to a certain extent against SARS-CoV-2. And that's because when you have your immune system generally stimulated, you're actually better prepared to deal with even a new virus that comes in. So our overall immunity in the population is actually declining continually the more lockdowns that we do. So that's the second. The third thing is that there's lots of epidemiology studies that have been done here in BC with I remember seeing one study for the BC Women's and Children's Hospital, another one across Canada. Health Canada has been tracking this, and this is a phenomenon around the world. The younger you are, the more anxious and depressed that the whole COVID pandemic has made you. And when you're anxious and depressed, and by the way, females more than males, you're more likely to actually have a depression of your immune system itself. There's a strong correlate between anxiety and depression and reduction in immune protection. So you have a perfect storm of all these things happening, especially for younger people. And I think that's the reason why we're seeing more people that are younger now that are ending up in hospital than we did a year ago. The absolute numbers have increased, not just the relative amounts, the absolute numbers have increased. So this is very disconcerting. So the vaccinations, I do not personally think it's a good idea for younger people to get vaccinated. I think they're better off being infected. And at the same token, I think that with the restrictions that we've been doing, we're actually setting up our population for a great deal more destruction down the road. So this is a concern there. And I haven't even talked about the social economic inputs. The research has been shut down pretty much at universities for the last year and a half on all these other diseases. New medications and treatments and diagnostics aren't getting developed as quickly as they were before, and that's lives being lost. where if these were out there, they could help people. So there's so many factors at play here.
Everything you're saying is making a ton of sense to me, and I can see from the comments to the people out there as well. And you're, a very, very intricately and appreciative. We're appreciative of this. You're kind of laying out there exactly what our information research has been the best that we can do, is kind of leading us to very similar deductions that we're probably heading for something even worse down the road from some of these rushed things that they cannot tell us, that there's going to be a certain number of people that are going to have very adverse reactions. And in my line of work, that typically I probably hear more reports about that than actually the adverse reaction sites do. But it's people that don't know how to use the process. And I find, and this is something that I'd like to look into further down the road, because I didn't realize the inadequacies and the gaps in information that are out there for people to, or access to that information. And my own deductions, as well as many people out there too that are here looking for answers to these same questions, are finding the same thing. And the more we learn about this, the more concerned we are about the measures creating, again, bigger, worse storms of complications because of intervening with nature in a rushed way. So very, very appreciative of you sharing that with us. I don't want to take up all of your afternoon, so we've gone just over an hour here. What would you like to close out with for folks here, in what you're hoping to achieve out of, or what you'd like to see come out of this effort that you're putting out?
Well, two things. One is that I wish that the government had spent more attention into developing tests to determine whether a person actually has protection and immunity against the virus, than simply a genetic test to determine whether or not you actually have the virus at that moment. Because genetic tests are very restricted to, you have to actually be infected with the virus. You'll never know whether you were infected with it before from that test. And after you recover, you wouldn't know that you had it unless you had the test done. So we should have had more testing done with antibodies. And I hope that with the vaccines, that we have a better sense of whether or not people are actually protected, because there's this idea of vaccine passports, and that's just crazy. Because even if you've been vaccinated, it doesn't mean you're actually protected. And people who actually have had the virus already have protection, and there's no need to expose them to risks associated with these experimental vaccines. So the second point I want to make is that, we're involved with this Canadian COVID Care Alliance. It's a dedicated group of more than 50 doctors and our researchers from across the country have gotten together. Our goal is to try to get the straight facts, to basically, social media on the one hand has got a lot of information out there that's just completely off the mark. And then there is information from legitimate sources, which is important to adhere to, but some of it is still also questionable. And so the biggest thing is related to policy, and the policies that we make with the government and as individuals, we have to be informed with the right information, like the information that's accurate and evidence-based. So the objective of our group is to allow people to understand what the alternatives are. I haven't talked, and I'm sure my colleagues will later, about some of the treatments that are available that currently are not available really to Canadians that easily, but are used effectively in other countries. And the importance of just maintaining your general health. The best way to keep your immune system really fully functional is to go out there and get exposed to things. Be healthy, eat good food. Well, there's very little discussion about the reduction of obesity and its huge impact. If you look at the people that are in these IC units in the United States, almost every one of them are obese. I mean, good food, good diet. There's so many things that you can do to protect yourself, and they're available to you. So we want to arm you with the information, where to go, where you can get this information, things that you can do. And we think we can save more lives this way. And that's really the objective of this organization, which, by the way, is we'll be launching our website shortly. And everybody who's been involved in this has done it really out of volunteer. We're not paid to do this. Most of us have decided to do this because it's really important to do. And we have these experiences and abilities and opportunities, and so we're trying to make the most of it for our fellow Canadians.
Well, again, there's no way that I can thank you enough on behalf of our community here on what you're doing, and I'm sure there's something different that compels each one of you into taking part in this. Do you see this growing in discussion? I know I've been hearing from people that have been hesitant to engage in these kinds of things like that. Is that something that you're seeing a shift in the discussion out there?
Well, one thing about science is that it always seeks the truth, and in time, sometimes it's not convenient. It takes longer than you'd like, but we'll start to better understand what's actually going on, and become more effective in dealing with this kind of a problem. So this has been a learning lesson for, I think, the community, the scientific community, as well as politicians. I think that for people, it's hard for people because they want to be good citizens. They want to protect other people. So people wear masks, but you have to not operate out of fear and paranoia. You have to put things into perspective and what the risks are to yourself and to your community, your family. And when I try to look at it in a scientific perspective, which is usually the best way to proceed when you're doing things, we just don't seem to be going down that route. Our politicians are being driven by their constituents, which are fearful and paranoid because of all the information that's been pumped to them. And let's face it, if it bleeds, it reads. So there's a good kind of incentive for a lot of the mainstream media to go ahead and publish information that follows a narrative. And they're dependent on their funding from government and from advertisers. A lot of them are pharmaceutical companies. So it makes one really a little bit suspicious about everything. And I understand that kind of concern about, first, if you're against vaccines. I'm not against vaccines. I just think that we can have better vaccines, and that we have to take what we have and use it the most effectively for those that are at greatest risk, but not subject those that are actually at low risk to the risks of the vaccine being much bigger problem necessarily than the virus itself.
Well, I think in Canada's case, and probably in many other nations, we have a society that is not only fairly well educated, but fairly adept at doing its own research and its own homework. And they're seeing a distinct difference in what they're finding on their own time versus what they're hearing from our government. And this is creating a deeper divide in what is happening, and people are feeling cornered in some of this. And so it's again so important in what you're doing in speaking up with this and creating meaningful dialogue. And this, you've shared so much with us today. Grateful for that, and hopefully we'll get a chance to do this again sometime, because what you're doing is absolutely amazing, and many kudos to you for that.
Well, thank you very much, Wayne. I really appreciate the opportunity to talk to your audience.
Well, it's. I hope you enjoyed yourself, and we didn't scare you away from this. We are not like the most of the mainstream media out there, and that's the thing.
No, I'm lucky. I'm coming up to 64, and I've got a full professor, and I have tenure, So I'm sort of the one that can come out there and talk more openly because I'm a little bit more insulated than many of my
Yeah.
colleagues. So
Well, one thing that I've learned in my journey in doing this, I was not a professionally trained journalist or anything. Absolutely, quite the contrary. But I was driven to do what I was doing, what I do now, because of those same things. What I was seeing, what I was learning, was not what I was hearing, and I felt compelled to do something about that and to spread this kind of truth. And I've seen that courage begets courage, and when there are enough courageous people, the truth cannot be hidden. So I'm very excited about what the future brings, and we can do things better once we get to the heart of the matter. So again, thank you so much for your time, and I will eagerly be following your work. I've learned so much about what you do today. This has been very entertaining and enlightening, and we'll keep you posted on how this goes. And
thank you.
but I suspect that there's because this is all based on a kind of a connection that we've made, without actually searching for each other through, you know, uh, like-minded activity. There's a scheduled discussion coming up real soon. I'm hearing that we may see your input in as well. So this is something we'll be talking about a little bit further on in the future. So I'm sure that the folks out here in What's Up Canada land, and for those that are out there watching, it's quite an international community. Eyes are on Canada, and you're leading the way. So we will we'll save lives with what we're doing. So
Thank you. Yep.
my pleasure.
Bye, everyone.
Bye for now. Wow, guys, that was a lot of fun. I hope you guys enjoyed that. I got a lot out of it. You guys know what to do from here. There's an immense amount of information coming. We're going to be trying to do this every day now for at least the next 10 days, bringing different people to do the same thing for you guys. So same time, same place tomorrow. And we'll keep you posted. We will try and get this stuff up onto Rumble and some of the other platforms as soon as we can, because we know what the social media platforms are doing to us. So you guys know what to do. Stay Canadian, and we will make Canada Canada again. Bye for now.